In this video, we will discuss about the role of Granzyme in cognition effect. So in the previous video, we have understood that Granzyme are found to be high in amount where this CD8 plus T cells adaptive transfer is done. So now what they did is they also check this Granzyme amount in the plasma of the aged CD8 plus T cells adaptive transfer cells and the young CD8 plus T cells adaptive transfer cells into the mice, and they are able to see that the aged CD8 plus T cells treated mice has high Granzyme in the plasma, and when this aged CD8 plus T cells treated mice is treated with the PTX, they are able to see reductions in the Granzyme, indicating it's the CD8 that is the main cause, or that is the main contributor to this increased Granzyme levels in the plasma of this aged CD8 plus T cells adaptive transfer mice. Afterwards, to find out the locations of this exogenous Granzyme, they integrated this Granzyme with HiBiT, which when interact with this LgBiT will give a luminescence signals. So from that they are able to see the location of this exogenous Granzyme in the mice. And when they did their studies, they are able to see that only in the fat case or in the periphery, they are able to see circulations of this exogenous Granzyme, whereas in the cortex and hippocampus they do not see significant changes between the control, that is GFP, or the Granzyme. So they only see the changes in the exogenous part, or they only see changes in this periphery or in the fat region. Afterwards, they wanted to see whether this Granzyme uh, will have any impact on this cognitions functions or not. So they did this Granzyme, they adaptive transfer this Granzyme or GFP, which is control, in the tail of the mouse, and then did the RAWM analysis and also this hippocampus dissections. So in the RAWM analysis, they are able to see that the mice that are treated with the Granzyme has more error compared to the the mice that are treated with the GFP, which is the control, indicating that the Granzyme has a role towards weakening cognition functions. Afterwards, they wanted to find out what is going on at the transcription levels. So for that, they take the uh, previously published putative Granzyme data and overlaid it with the single cell RNA-seq data of hippocampus. And from that, they are able to find out that these Granzymes are found to be overexpressed in the vascular populations, endothelial populations, and the other related populations. Whereas, surprisingly, they see a low level of Granzyme in the neural populations, especially in these accelerated neuron populations. Afterwards, when they compare these Granzyme putative targets, which are previously published, along with this differentially expressed gene in the aged CD8 plus T cells adaptive transfer into the mice, they are able to find out that 46 genes are common in between, whereas so these 46 genes they later on used to find out what are the common pathways that are dysregulated in the case of this DEG, differentially expressed gene in the aged CD8 plus T cells adaptive transfer, and the Granzyme putative target. This will give us the common idea or common pathways that shows the Granzyme that are overexpressed by CD8 plus T cells cause this cognition weakening, cognition decline, because the CD8 plus T cells has a direct role with this increase in the Granzyme that is shown in the panel H. And by analyzing and this Granzyme pathways and the CD8 plus T cells dysregulated gives some common pathways. These common genes could give us an idea about the pathways that will cause because of both CD8 plus T cells and Granzyme. Mainly, the CD8 plus T cells will cause increase in the Granzyme, and those increase in Granzyme will cause these changes in the pathways. We will discuss those gene ontology analysis of these common genes in our subsequent videos. However, from this video, I hope you understood the from this video the results. I hope you understood the results that are presented in this video. That is about the connections between CD8 plus T cells and the granzyme. And the granzymes locations in different parts, and the transcription changes that occurs because of the overexpression of the granzyme. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.